I was out walking earlier today, just trying to clear my head before we sat down to record.
Oh, nice. Good day for it.
Yeah, it really was. But I actually found myself stopping in front of this absolutely massive oak tree.
Okay.
And I don't just mean glancing at it as I walked by. I mean really stopping and just staring at the sheer architectural scale of the thing.
It's easy to overlook when you see them every day.
Exactly. I was looking at the way the bark fractured into these deep, rugged plates, and then tracing the trunk all the way up to the canopy, looking at the thousands of delicate little leaves catching the sunlight.
Right.
And I started thinking about the root system, too, which is entirely invisible to us, but probably spreads out just as wide underground.
Usually even wider, yeah.
Which is crazy to think about. But as I stood there, it hit me in a way it never quite has before.
What did?
The origin of it all. Every single distinct part of that giant, complex, multi-ton structure originated from one single microscopic speck.
One fertilized egg.
Yes, a zygote. I mean, the tough bark, the fragile flowers, the deep roots, the vascular plumbing that pulls water hundreds of feet into the air. All of it. All the instructions, all the potential for that immense physical structure were somehow packed into an invisible starting point.
It really is the ultimate biological marvel.
It is. It's a profound realization when you really sit with the mechanics of how that is even biologically possible.
And honestly, that sense of scale, that sense of origin, is exactly the mindset you need for this topic.
I was hoping it would tie in.
Oh, perfectly.
Wow.
In my 15-plus years mentoring biology students, especially the ones preparing for incredibly rigorous, high-stakes medical entrance exams.
Like Neet.
Exactly, like the Neet over in India, which tests absolute conceptual clarity.
Yeah.
This is the exact breakthrough moment I wait for with a student.
Really, the tree moment.
The tree moment. It's the moment a learner transitions from just, memorizing vocabulary on a page to actually visualizing the staggering reality of the biology.
Because it stops being just text.
Right. That realization you just had under the oak tree, that a microscopic zygote can meticulously construct a towering organism over decades without the entire system just devolving into chaos. That is the foundation of everything we're exploring today. You've essentially stumbled right onto the core mystery of plant biology.
I am so glad that awe translates, because it feels like something we take entirely for granted. You see a tree, you know it came from a seed, but you don't really think about the engineering required to get from point A to point B.
We completely skip over the how.
So for you listening right now, welcome to this deep dive. Today, we are taking a massive step back from the micro details to look at the macro picture.
The big picture.
Exactly. We are diving deep into the fundamental principles of plant growth and development. Our mission today is to extract the core logic of how a plant builds itself from scratch.
Because it's a highly ordered sequence of events.
We want to set up a rock-solid conceptual foundation for you before you even think about tackling the heavier, highly technical chapters.
The plant physiology, cellular respiration, photosynthesis.
Right. Before all of that, you need to understand the blueprint. We're going to build your mental framework first, so that all those complex biochemical pathways actually have a structural context to live in.
And that structural context is precisely what separates top-tier understanding from just superficial memorization.
It makes everything else stick.
It really does. We are looking at the introductory principles today, the foundational text that sets the stage for the entire field of plant physiology.
But people tend to skip the intro, don't they?
All the time. It is such a common trap for learners to gloss over these opening concepts. They want to rush past the introduction to get to the meat of the subject.
The complex diagrams and the chemical formulas.
Exactly, the hormonal pathways. But what's so fascinating and frankly crucial is that these introductory concepts contain the master blueprint.
The fundamental logic.
Right. If you do not understand the underlying why and how of development introduced here, the rest of the technical details you learn later will just be disconnected facts floating in your head. To master the biology, you first need to understand the fundamental logic of the plant's design.
Let's cut straight to the essence of that master blueprint then.
Let's do it.
The core idea running through our source material is that plant development is not just some random explosion of cellular division.
Not at all.
It is not a chaotic ballooning of biological tissue. The text explicitly defines development as a precise and highly ordered succession of events.
Precise and highly ordered.
Right. So to really visualize what that means, I want you to imagine a 3D printer.
Okay, I like this.
But not just a standard plastic extruding machine on a desk. Imagine a microscopic, autonomous 3D printer that you drop onto a patch of soil.
Just drop it in the dirt.
Yeah. and it starts printing a massive factory. That's the main plant body. But it doesn't just print the structural walls, it also prints its own solar panels, the leaves, to power its own construction. It prints its own internal plumbing system to move water and nutrients. It prints its own reproductive products, the flowers and fruits.
And it does it all on a schedule.
Yes, that's where this biological printer becomes truly mind-bending. It knows exactly when to shed old parts, like dropping leaves in the autumn to conserve energy.
And exactly when to put new ones in the spring.
Exactly. And it manages this entire dynamically shifting construction project from that single initial speck of genetic material, perfectly timing every single step over the course of months, years, or even centuries.
Taking that 3D printer analogy a step further, think about the materials it actually uses.
Oh, right, because it doesn't have a plastic spool.
Exactly. A normal printer has a cartridge of ink or plastic provided for it. This biological printer is pulling invisible carbon dioxide gas out of the air,
Wow.
pulling liquid water up from the dirt, and using the radiation from a star 93 million miles away to physically synthesize its own building materials.
That is just an extraordinary feat of self-assembly.
It is, and looking at it this way raises an incredibly important biological question. Why do the strict rules of development even exist?
Right. What specific problem is nature solving here?
Well, consider the alternative scenario. If cells simply divided endlessly, just metabolizing and replicating, without a strict overarching master plan,
No, what happened?
you would never get an oak tree. You would just get a featureless, chaotic mass of biomass.
A giant green blob.
A giant green blob. And in fact, we see this in nature occasionally.
We do.
Yes. When certain bacteria infect a plant and disrupt its developmental genetics, the plant produces what is called a crown gall.
Crown gall.
Yes, essentially a plant tumor. It is just a disorganized, unspecialized mass of dividing tissue.
Oh, wow. So it's just raw growth without the instructions.
Exactly. The entire field of plant growth and development exists to explain how biological chaos is reined in. It explains the mechanisms that organize raw cellular division into structural and functional perfection.
How the zygote dictates the sequence.
Precisely. It is the story of how a single zygote dictates the highly deliberate, unyielding sequence that gives rise to roots, stems, leaves, flowers, fruits, and seeds.
A featureless green blob. That paints such a vivid picture of what happens when the rules of development are removed. Without that precise and ordered succession, it's just uncontrolled, pointless expansion.
Pure chaos.
Which brings us perfectly to the guide we need to map out this journey. If we are going to understand how the plant avoids becoming that chaotic green mass, we need a mental map of the rules it follows.
The foundational equation.
Exactly. Our source material provides what is arguably the most critical foundational equation in this entire branch of biology.
It's the core of it all.
It is elegantly simple but profoundly important. Here it is. Development equals growth plus differentiation.
If you want to connect everything you learn in plant biology to a single unifying concept, that equation development equals growth plus differentiation is the lens through which you must view every single process.
So let's really break those two terms down.
We have to. Because a very common conceptual hurdle, even for advanced students, is treating those words as synonyms.
Yeah, in everyday conversation, we might say a child is growing or developing, and we mean the exact same thing.
Right. But in biology, precision is everything. Growth is strictly defined as an irreversible, permanent increase in size, mass, or volume.
It's the physical expansion.
Exactly. At a cellular level, this means cells are dividing to increase in number and then physically stretching and expanding.
How do they stretch?
Usually by taking in massive amounts of water into their central vacuoles to literally push against the cell walls.
Ah, so they inflate.
Essentially, yes. But if a plant only possessed the ability to grow, we are right back to the featureless green blob scenario.
Because they're just getting bigger.
Right. You would just have a much larger mass of identical, unspecialized cells. They wouldn't be doing anything useful.
Which is exactly why the second half of that equation is so vital. Differentiation.
The game changer.
If growth is the act of getting physically bigger, differentiation is the act of getting specialized. It is the process of a generic cell getting a highly specific job.
Finding its purpose.
So a cell stops merely dividing and expanding, and its internal genetic programming says, all right, my dividing days are over. I'm going to become a rugged bark cell to protect the trunk.
Or I'm going to become a fragile, pigmented flower petal cell to attract a pollinator.
Exactly. It fundamentally changes its physical structure, its internal chemistry, and its cell wall composition to perfectly fit a specific function.
So you need both.
You absolutely need both elements to build a functional organism. You need the raw physical materials and the expansion of size, which is growth, and you need the complex specialization of those raw materials, which is differentiation.
And when you add those
You
together,
add those two distinct processes together, and the sum total is true biological development.
that is exactly the distinction learners need to internalize. And this mathematical sum of growth and differentiation doesn't just happen all at once, randomly.
No, it has a timeline.
Right. It unfolds across a very strict life cycle progression, which is beautifully detailed in our source text.
Yeah.
Let's trace that exact sequence, because building an intuition for this timeline makes all the subsequent physiology much easier to grasp.
The journey starts with the zygote, right? Like with the oak tree.
Exactly. The journey starts with the zygote, the single fertilized egg resulting from sexual reproduction. Through initial rounds of division, that zygote develops into an embryo housed inside a seed.
The survival capsule.
A perfect way to describe it. The seed is essentially a survival capsule, holding the developmental process in stasis until the environmental conditions are perfectly aligned.
And
Then, upon germination, we get the seedling. The text explicitly points to the visual example of a germinating bean. You can actually see the first root, the radical break through the seed coat.
followed by the shoot pushing up toward the light.
Yes, and from that vulnerable seedling stage, it establishes itself into a plantlet. And finally, through years of sustained growth and differentiation, it becomes a mature plant capable of producing its own zygotes.
Zygote, seed, seedling, plantlet, mature plant.
It is a highly ordered, one-way directional progression of life.
I find that progression so satisfying to think about because it's something you can literally observe in one of those beautiful time-lapse nature videos.
Oh, those are the best.
You watch the seed crack, the root dive down, the shoot reach up. But as the plant progresses through those later stages, reaching maturity, it experiences a phenomenon the text highlights that I found completely fascinating.
The two clocks.
Yes. It is a wild biological paradox that I like to think of as the two clocks of plant growth.
It's a great concept. Let's unpack it.
So clock number 1 is indeterminate or continuous. The text points out that trees and many plants continue to increase in height or girth over a massive period of time.
Pretty much forever.
Right. The apical meristems at the tips of the branches keep reaching higher, and the lateral meristems inside the trunk keep packing on wider rings of wood. They essentially keep growing for as long as the organism is alive.
That's the continuous clock.
But simultaneously, you have clock number 2, which is determinate or periodic. The leaves, the flowers, the fruits, these organs have strictly limited dimensions.
They don't just grow forever.
No, a leaf only grows to a specific genetic size and then it stops completely. And beyond just stopping, these organs appear, age, and fall off periodically, often repeating this cycle year after year.
It seems like a contradiction, doesn't it?
It totally does. How does a single unified organism operate its biology on two completely different contradictory growth clocks at the exact same time?
It seems contradictory until you view it through the lens of evolutionary strategy and resource management.
Okay, make it make sense for me.
The logic behind running two distinct growth programs is purely about long-term survival in a fluctuating environment. Let's look at it from a structural engineering perspective.
Like the factory again.
Exactly. The main body of the plant, the central trunk, the major scaffolding branches, the deep anchoring root system. This is the foundational infrastructure. It must continuously grow to support an ever-increasing demand.
Because it's getting bigger and heavier.
Right. As the tree gets taller to outcompete its neighbors for sunlight, it needs a wider, stronger trunk to support the physical weight and resist wind shear. It needs a continuously expanding root system to find enough water to sustain that massive canopy.
So the continuous indeterminate growth is non-negotiable for structural stability.
Non-negotiable. That is your first clock. However, the organs operating on the second periodic clock, the leaves, the flowers, the fruits, these are highly specialized, energy-intensive components.
The workhorses and the reproductive machinery.
Yes. Because they are so specialized and exposed, they are incredibly vulnerable to environmental extremes, like freezing winter temperatures, severe drought, or insect infestations.
So instead of trying to armor a delicate leaf against a blizzard, it makes far more biological sense to just cut your losses and replace it later.
Precisely. Why would a tree expend massive amounts of precious metabolic energy trying to keep a fragile, water-filled leaf alive through 3 months of freezing winter darkness.
It's a losing battle.
It is. Instead, the plant utilizes that second periodic clock. It systematically shuts down the leaf, salvages all the valuable nutrients back into the prominent trunk, sheds the useless structure, and goes dormant.
It conserves its core energy in the main infrastructure.
Then, when conditions improve in the spring, it utilizes its continuous growth regions to simply print brand new, highly efficient, undamaged leaves.
That is so smart.
The cyclical, replaceable nature of these determinate organs allows the plant to dynamically adapt to shifting seasons and constantly renew its energy-gathering machinery without having to rebuild its entire body from scratch every single year.
It is a masterclass in the efficient allocation of biological resources.
Truly.
It really is like keeping the heavy factory walls intact, but constantly upgrading and replacing the delicate machinery inside based on the season.
That's the perfect analogy.
And speaking of factory logistics, that perfectly segues into another major developmental rule the text brings up, the phase shift rule.
The sequence of phases.
Yeah, the text poses a very direct, probing question to the reader. It asks, why does the vegetative phase precede flowering in a plant?
A great question for learners to ponder.
And if you think about it logically, based on what we just discussed with the factory analogy and resource management, the sequence makes perfect sense.
How so?
You absolutely have to build the foundational factory, the vegetative tissues, meaning the extensive root networks, the sturdy stems, and a massive canopy of leaves before you have the stored energy, the structural support, and the surplus resources required to produce the luxury goods.
Which are the flowers, the nectar, and the seeds.
Exactly. You simply cannot expect a biological 3D printer to output a complex final product if it hasn't even finished printing its own power supply and plumbing yet.
That is the harsh biological reality of energy economics. The vegetative phase of a plant's life is entirely about accumulation.
Hoarding resources.
Yes. It is the phase where the plant is aggressively building its solar panels to capture light, establishing its deep water pumps, and creating a sturdy structural framework.
It is hoarding carbohydrates and minerals.
And reproduction is the opposite of hoarding.
Reproduction, the act of flowering, producing pollen, generating nectar to attract pollinators, and finally packing dense, nutrient-rich endosperm into seeds, is an astronomically expensive process biologically.
It's a massive drain.
It drains immense amounts of energy and structural resources away from the rest of the plant. If a plant attempted to trigger its flowering phase before it had established a robust, mature vegetative system to support that massive energy draw,
That would just crash.
it would literally exhaust its reserves. It would likely die of starvation or structural failure before the seeds could even fully mature.
Wow.
Therefore, the ordered sequence of development strictly dictates that infrastructure, accumulation, and basic survival must definitively precede the luxury of reproduction. I want to pivot our focus here because understanding these beautiful logical systems is incredibly rewarding. But I know many of our listeners are actively applying this knowledge in highly competitive academic environments.
The exams.
The exams. Knowing the biological concepts is only half the battle. Knowing exactly how those concepts will be tested, twisted, and probed by examiners is the other critical half.
It's a completely different skill set.
So let's lean heavily into your 15 years of direct mentoring experience. When a seasoned examiner looks at this seemingly simple, foundational, introductory text about plant development, what specific concepts are they extracting to build difficult questions?
I am thrilled we are dedicating time to this.
Where do students consistently lose marks? What is the high-yield warning we need to issue right now?
Because this introductory material is exactly where overconfident students lose the easiest marks. Examiners absolutely love introductory paragraphs because they know human nature.
They know students skip ahead.
They know students tend to skim the first few pages to get to the complicated diagrams later in the chapter. So let's start with a massive hurdle. Let's look at a classic conceptual trap regarding the fundamental equation we discussed.
Development equals growth plus differentiation.
Exactly. Examiners will constantly try to trick you by mixing up the precise definitions of these three specific terms. They will present a multiple-choice question or an assertion reasoning statement where they define development as merely an irreversible increase in physical size or mass.
And if you're rushing, that sounds correct.
It sounds completely fine, but that is actually the strict definition of growth. Or they will use development and growth interchangeably in a sentence to see if you notice the error.
Because they are not synonyms.
You must absolutely lock into your brain that development is the sum total of two very distinct separate processes. If a mass of plant tissue expands and grows but never differentiates into specialized cells, it is not fully developed.
It's just a bigger blob.
Right. Conversely, if a cell alters its internal structure to differentiate but doesn't undergo the volumetric expansion of growth, the tissue hasn't fully developed either. You fundamentally require both expansion and specialization to achieve development, recognizing that distinction is an easy way to secure points.
Okay, so the first major trap is treating the equation like a list of synonyms rather than a strict mathematical biological reality. Don't mix up expansion with specialization.
Exactly.
What is another area where people get tripped up by the wording?
Another absolute classic involves what I call the rule of the universal ancestor.
The universal ancestor.
Yes, there is a very specific definitive sentence in our source text. All cells of a plant are descendants of the zygote.
Which makes sense based on our oak tree realization.
It does. Now, examiners love to turn this into tricky true-false statements or complex assertion-reason questions because they prey on a student's advanced knowledge.
How does advanced knowledge hurt you?
As you study more advanced biology, you get conditioned to look for exceptions.
Mm.
You learn that in biology, there is almost always a weird edge case. So an examiner might frame a statement like, most cells of a mature woody plant are descendants of the zygote, with the exception of the heavily lignified dead vascular tissues.
Oh, that sounds so plausible.
Right. Or they might throw in a distractor about cork cells. Students sitting in the exam hall start overthinking. They think, well, cork is dead bark, maybe it forms from environmental calcification. Or surely the hollow water pipes don't come from the same place as the living leaves.
They talk themselves out of the right answer.
Exactly. Let me be unequivocally clear, and you need to trust this. In the natural development of a standard seed plant, there are 0 exceptions.
0.
Every single cell, whether it is a completely dead, hollow wood cell in the deep center of an ancient trunk, or a vibrant, living, metabolically active cell in a freshly opened petal, traces its cellular lineage through thousands of divisions directly back to that one single fertilized egg.
One zygote rules them all.
It is an absolute unbroken rule of developmental lineage, and examiners will ruthlessly test your confidence in that absolute rule by offering you fake exceptions.
That is genuinely a brutal trap because you are so right. The deeper you get into science, the more you expect every rule to have an asterisk next to it.
But not this one.
Here, the rule is the rule. And speaking of where the syllabus goes from here, the introductory text gives us a literal roadmap of the upcoming highly technical subtopics.
It lists out the exact sections the student's about to face.
Right. It drops terms like growth, differentiation, dedifferentiation, redifferentiation, plant growth regulators, photoperiodism vernalization. From a pure high-yield exam preparation perspective, why do examiners care so much about these specific heavy-hitting keywords? Why should you care about these terms right now?
Because these specific keywords represent the heaviest scoring areas in all the subsequent plant physiology chapters. Understanding their root meaning and conceptual purpose right now will save you countless hours of confusion and memorization later.
So let's look closely at the differentiation family of words, because you mentioned this trio gets tested constantly.
Constantly. You have basic differentiation, which we already defined as a generic cell getting its specialized job, like a cell becoming a protective bark cell.
Okay, that one is clear.
But then the syllabus introduces dedifferentiation and redifferentiation.
That's so complicated.
The prefixes actually tell you the entire biological story. D means to undo or reverse. So dedifferentiation is a fascinating process where an already specialized mature cell somehow forgets its highly specific job.
It forgets.
It sheds its specialization and reverts back into a generic, actively dividing stem cell-like state.
Why on earth would it do that?
Usually for wound healing. If a tree trunk is gashed open, the nearby mature cells dedifferentiate so they can rapidly divide and form a mass of tissue to cover the wound.
That is incredible. They hit the reset button.
Exactly. Then you have which means to do again. Once that wound is covered by generic dividing cells, those new cells must redifferentiate. They must take on a new, different specialized job, perhaps becoming scar tissue or new vascular cambium.
So they specialize, unspecialize, and then respecialize.
You've got it. Examiners will endlessly test your ability to track a specific plant cell's journey through those 3 distinct phases. They will ask you to identify which stage a cell is in during secondary growth or wound repair.
Okay, that makes so much sense now. And what about the other major terms? The text mentions that this entire complex process of development is governed by control factors that are broadly categorized as intrinsic and extrinsic, internal and external. I imagine that division is a massive framing device for how the rest of the unit is taught and tested.
Absolutely. It is the fundamental dichotomy of plant biological control. Everything a plant does is a reaction to either an internal signal or an external environment.
Internal versus external.
Right. Intrinsic factors are internal controls. These are the plant's own inherited genetics and its own internal chemical messengers. This is where the massive, highly tested topic of plant growth regulators comes in.
The hormones.
The plant hormones, auxins, gibberellins, cytokinins, abscisic acid, ethylene. These are the chemical signals the plant uses to talk to itself, to coordinate growth between the roots and the shoots.
And the extrinsic factors.
Extrinsic factors are external to the surrounding environment. And the text points to two highly specific, heavily tested environmental triggers: photoperiodism and vernalization.
Break this down for me.
Photoperiodism is how the plant reacts to the relative length of day and night. It is essentially how a plant measures the time of year using light-sensitive proteins called phytochromes.
So it's basically a light clock.
Exactly. And vernalization is how the plant reacts to prolonged periods of cold temperatures, essentially using the memory of winter to ensure it only flowers in the safety of spring.
The memory of winter, that's poetic.
In an exam setting, examiners will present a complex biological scenario and ask you to categorize the driving phenomena. For instance, if a plant only flowers when the nights are longer than a critical duration, is that triggered by an intrinsic or extrinsic factor?
It's extrinsic, driven by the environmental cue of photoperiodism.
Spot on. Understanding this strict, logical division between internal hormonal control and external environmental influence is absolutely crucial for navigating the complex questions later on.
It is so helpful to have that roadmap laid out clearly. It takes a lot of the fear out of those big, intimidating words when you realize they are just describing how a plant heals a wound or how it knows when it's winter.
Exactly. They're just logical processes.
But I want to zoom back out for a second. While understanding the exam focus and the terminology is critical for you as a learner, the sheer elegance of the underlying biology here is just completely mind-blowing to me.
It really is.
We touched on this briefly earlier, but I want to dive really deep into a concept I call the identical origin paradox.
Okay, lay it on me.
If, as we firmly established, every single cell in that massive oak tree is a direct descendant of the exact same initial zygote.
With zero exceptions.
With zero exceptions. That means every single cell in the tree has the exact same genetic starting point. They all possess the exact same DNA sequence.
Correct. Genomic equivalence.
Right. The tough, dead, lignified bark cell, the microscopic root hair absorbing water, and the soft, vibrant, fragrant petal of the flower are all genetic clones. They have the identical genomic map.
They do.
The profound mystery, which our source text asks directly, challenging you to really think about it, is why and how do these genetically identical siblings develop completely different structural and functional attributes?
It's the ultimate question.
How is it physically possible for them to have wildly different destinies and shapes if they're all reading from the exact same architectural blueprint?
What is so fascinating about your question is that you are touching upon the central animating mystery of all developmental biology, not just in plants, but in humans as well.
It applies to us, too.
Oh, absolutely. If the genetic instruction manual is perfectly identical in every single cell,
Right.
how do you get such drastically different physical outcomes?
Right. How?
The answer, without getting too deeply bogged down in the molecular biochemistry, lies in how the individual cells read that manual.
They read it differently.
While every single cell possesses the entire unabridged book of the plant's DNA, they do not all read the same chapters.
Oh, I see.
Depending on their physical position within the developing plant structure and the specific cocktail of chemical signals they receive from their neighboring cells, those intrinsic hormonal factors we just talked about, different cells are instructed to turn on certain specific genes and completely silence others.
So location dictates their destiny.
Through complex positional information, often driven by gradients of hormones like auxin, a cell on the outer layer of the stem receives a signal that says, "You are on the boundary."
And how does it react?
That cell responds by opening the chapter on protection. It turns on the produce tough lignin and suberin genes to become bark. Meanwhile, a cell in the floral meristem receives a completely different chemical gradient.
So it opens a different chapter.
Exactly. It is prompted to silence the bark genes and turn on the produce colorful anthocyanin pigment genes to become a petal.
That is just an incredibly elegant system.
It is a highly coordinated, flawlessly timed symphony of selective gene expression. The external environment and the internal chemical signals tell each individual cell which specific part of its identical blueprint to actually execute.
They all own the entire encyclopedia, but they only ever open the one specific volume that corresponds to their physical location and job.
It's brilliant.
It is nature at its most efficient.
And speaking of the arc of a plant's life, there is another realization I had reading this that I found surprisingly philosophical for a dry biology text.
Biology can be very philosophical. What was the realization? The text outlines this beautiful, highly ordered progression we've been talking about. The zygote dividing, forming a complex structural body, producing roots, expanding leaves, growing branches, ultimately creating flowers, fruits, and seeds to pass on its genetic legacy.
The whole sequence.
Right. And then right at the end of that triumphant list, it drops this incredibly blunt phrase, and eventually they die.
Ah.
It states it so matter-of-factly. But what struck me is that death isn't framed as a failure of the plant. It's not framed as a mistake or a disease or something going wrong. It is literally listed as the final expected step in a precise and highly ordered succession of events.
It's part of the plan.
Planned obsolescence, a programmed end, is built right into the fundamental biology of the organism.
It requires a profound shift in perspective to truly grasp that concept. As humans, we almost universally view death, especially in medical or biological terms, as a failure of the system.
A breakdown.
A breakdown, succumbing to entropy or disease. But in the grand context of plant development, death is very frequently a highly regulated, intentionally programmed, and deeply necessary event. In biology, we call this programmed cell death, or on a larger organ scale, senescence.
Senescence.
Think back to the leaves falling periodically that we discussed earlier. The death of that leaf in the autumn is not it's simply freezing and rotting away.
It's active.
It is an active, highly ordered, genetically controlled process where the plant intentionally breaks down the leaf's chlorophyll, which is why we see the beautiful autumn colors of the other pigments.
Oh, wow.
Salvages all the valuable nitrogen, phosphorus, and carbohydrates, pulls them back into the safety of the main trunk, and then cleanly surgically seals off the vascular connection, allowing the dead, empty husk of the leaf to fall.
So it strips the house of valuables before tearing it down.
Exactly. Furthermore, the very structural integrity of the tree depends on death.
What do you mean?
The xylem, the vast network of microscopic pipes that pull water from the deep roots all the way up to the canopy, is entirely composed of dead cells.
Wait, the water pipes are dead tissue.
Entirely dead. For those cells to function as hollow pipes, they must grow, differentiate, build incredibly thick, tough walls, and then intentionally trigger their own death to hollow themselves out.
They kill themselves to become a pipe.
Their death is their ultimate functional purpose. Even the death of an entire whole plant, like an annual crop, say a wheat plant, that dies immediately after producing its grain, is genetically programmed.
It just finishes its job and checks out.
The plant has successfully fulfilled its ultimate biological imperative, reproduction. Its programmed death rapidly clears the physical space, and its decaying biomass provides the rich organic matter necessary for the very seeds it just dropped to thrive in the next season.
It is the ultimate confirmation that the entire life cycle is not just a random struggle to stay alive.
No, it is a meticulously ordered succession from the very first division of the single zygote to the final purposeful programmed decay.
The water pipes of the tree are made of dead cells that killed themselves specifically to become pipes. That is going to stay with me for a long time. It really is a complete, perfectly managed cycle.
Biology leaves nothing to waste.
All right, we have covered a massive amount of conceptual ground today. We have moved from the visualization of autonomous 3D printers through the strict equations of development, navigated the heavy-hitting exam traps of dedifferentiation and identical origins, and ended up discussing the biological necessity of programmed death.
A very deep dive indeed.
For you listening, whether you are prepping for a massive exam or just expanding your understanding of the natural world, we want to make sure the absolute core logic of this deep dive sticks with you permanently. So let's lock in our final memory anchors.
This is the crucial synthesis. If you take nothing else away from this extensive discussion, I want you to mentally engrave these 5 critical facts into your conceptual framework.
Let's do it.
Here's what you must remember. Fact number 1. The master equation. Development is the sum total of growth plus differentiation. You absolutely must have both the physical size increase and the functional cellular specialization to achieve true development. They are not synonyms.
Growth plus differentiation. Got it.
Here is what you must remember. Fact number 2, the ultimate ancestor. The microscopic zygote is the single sole source of every single cell in the entire mature plant body. In a natural seed plant, there are 0 exceptions to this lineage.
No exceptions. One zygote rules them all.
Here is what you must remember. Fact number 3, the dual growth pattern. Plants operate simultaneously on 2 different clocks. They utilize indeterminate continuous growth for their main permanent infrastructure, increasing their height and girth. But they employ determinate, limited, periodic growth for their highly specialized replaceable organs like leaves, flowers, and fruits.
The factory walls versus the machinery inside. I have the last 2. Here's what you must remember. Fact number 4. The strict developmental sequence. The vegetative phase of accumulation and infrastructure building absolutely must precede the reproductive flowering phase. You simply have to build the factory and secure the energy before you can produce the biologically expensive luxury goods.
Perfect. And the final fact.
Here's what you must remember. Fact number 5. The 2 controllers of development. Every single step of this intricate process is dictated by 2 distinct camps of factors. Intrinsic factors, which are the internal chemical and genetic signals like plant hormones, and extrinsic factors, which are the external environmental cues like light duration and temperature shifts.
Excellent. If you internalize those 5 conceptual pillars, they will support every single complex piece of plant physiology you encounter in your future studies.
Okay, to seal the deal, let's do the rapid-fire sticky summary. The absolute essence of the topic in 60 seconds. Here we go.
The clock is ticking.
You start your journey as a single microscopic zygote. You undergo growth, which strictly means getting physically bigger, and you undergo differentiation, which means getting a highly specialized cellular job. Together, those 2 processes equal development. You follow a strict, unyielding, one-way sequence, from seed to seedling to plantlet to mature plant. You are forced to build your sturdy vegetative body first because you need massive amounts of stored energy to afford making your flowers and seeds later. As you age, you keep getting continuously taller and wider to compete, but you cycle through and replace your delicate leaves and fruits like a factory swapping out old machinery. And absolutely everything you do every single day is perfectly controlled by internal hormonal triggers from your own body and external pressures from your environment, right up until the highly ordered sequence finally comes to its purposeful, programmed end. Boom. The entire logic of plant growth and development in a nutshell.
A flawless summary. You have successfully distilled a massively complex biological symphony into its most essential logical movements.
As we finally wrap up this deep dive, we always like to leave you with a final provocative thought, something outside the textbook to turn over in your mind.
I love this part.
We've spent this entire time looking deeply at how a plant's entire life cycle, from the very first moment a root breaks out of a tightly packed seed to the exact genetically programmed moment a fragile autumn leaf falls to the ground, is highly ordered. We've seen how every phase is fundamentally shaped by intrinsic internal factors and extrinsic external factors.
So I challenge you to consider your own development as a learner and as a human being. Think about the mental map, the intellectual infrastructure you are building right now as you study. How much of your intellectual growth, your deep, lasting understanding of the world, is driven by your own internal, intrinsic curiosity, that pure inner drive to ask why and how, just for the sake of knowing?
And conversely, how much of your development is being shaped by the extrinsic pressures of your environment, the looming exams, the strict deadlines, the expectations of mentors or society?
Just as a plant fundamentally requires both its internal genetic instructions and the external sunlight to thrive and mature, understanding what truly drives your own learning development might be the ultimate key to mastering not just biology, but anything you set your mind to achieving.
That is something to seriously mull over the next time you sit down to study. Keep that mental map of the plant's journey handy as you continue exploring the biological world. And please remember to look a little closer the next time you walk past a towering tree. You are not just looking at wood and leaves. You are looking at the ultimate ongoing masterpiece of highly ordered development. Until next time, keep diving deep.